We'll now talk about the algorithmic problem of sorting by reversals. Let's start from a short chromosome consisting of just 10 blocks, and we'll number them in the order 1, 2, 3 to 10. Now, a reversal, you can think about a reversal as something that breaks a chromosome at two places and glue the resulting pieces in a new order as shown on this slide. And as a result, the reversal introduces two breakpoints, disruptions in gene order. For example, +3 and -8 are not supposed to be together, but they are together in the new permutation. And the same thing applies to -4 and +9. Let's take a look at one of possible rearrangement scenarios. In this case, transforming permutation into identity permutation, which is +1, +2, +8, with 5 reversal. Is it the shortest scenario? No, because there is a shortest scenario that transforms the same permutation into identity with just 4 reversals. And we'll define reversal distance as the minimum number of reversals to transform one permutation into another. Biologists are often interested in shortest scenarios for transforming one genome into another because this shortest scenario often correspond to biological scenario or come close to biological scenarios. So we now have transformation of one genome into another with 4 reversals, but maybe there is even faster transformations of these genomes with just 3 reversals. And reversal distance problem is the problem of calculating the reversal distance between 2 permutations, input 2 permutations, and output the reversal distance between them. We will also be talking about sorting by reversal problems, which is use as an input just a single permutation and ask for the reversal distance between this permutation and the identity permutation +1, +2, +n. Why identity permutations? Because we are at liberty to label the final permutations the way we want, and we will usually label this as +1, +2, +3, +n. Now, what would be the most natural algorithm for sorting by reversal? I'm not saying good algorithm, but the most natural one. Let's look at this mouse permutation for X chromosome in this case. +1 is already at the right position in this permutation, but +2 is not. However, we can use a reversal shown in bold here to bring +2 at the right position. That's what we've done right here. We brought +2 at the right position, but now it has a wrong sign. Not a problem. We will reverse the sign using a short reversal right here. Now, we placed both +1 and +2 at the right position, but -3 is at the wrong position. Not a problem. We'll find a reversal that brings it at the right position. Now, let's take care of +4 once again. Let's find reversals that brings it to the right position. It now has a wrong sign, but we will revert it. And we will continue doing this until we finally, using this greedy algorithm, transform mouse into Philip. And after we looked at the greedy algorithm for sorting by reversal, you may notice that, of course, it's not optimal. It used 11 reversals to transform mouse into human permutations, but we know from the first slide in this lecture that it can be done in just 7 reversals. So maybe we should come up with a better algorithm, and to do this, we will have to define the notion of breakpoint graph.